All right. Well, I'm Corey Anderson. I'm a student at Boise State University and taking the class Alien Worlds and Alien Life with Brian Jackson. Uh,
Sounds fun.
and yes, and then if you don't mind introducing yourself.
Okay. I'm Christopher Carr. I'm an assistant professor at Georgia Institute of Technology or Georgia Tech, and I'm in the aerospace engineering department. I have a dual appointment in Earth atmospheric sciences, and my group works on instrumentation for astrobiology and space biology, and we're seeking and supporting life beyond Earth.
Well, sweet. That goes in line with the class pretty good, and me and my group, they aren't able to join, but we were very interested in some of the work that was kind of we were interested in finding out more about your work as an engineer and biologist. Uh, so I guess our first question is what evidence do you look for when searching for life on exoplanets?
Well, that's a tough question. I personally am focused on the direct search for life in our solar system, as opposed to looking for life on exoplanets.
Mhm.
But I think we just heard about a recent study from using James Webb to look at uh, absorption lines that correlate with different gases and exoplanet atmospheres. And we're now starting to get a taste of what other worlds might be out there that might have liquid water. In this case, there was detection of carbon dioxide and methane, and the inference of an ocean world, possibly with a low reliability detection of DMS, which is only produced on Earth by life. And so we're now getting to the point where we are going to be exoplanets are a really great way to get high end because in our solar system there's a very limited number of different planetary bodies. But uh, exoplanets are kind of far away, even though there may be trillions of habitable exoplanets, and there may be statistically maybe a habitable world within, I don't know the right number, but maybe 10 light years, perhaps. That's still such a distance that we're not going to get there in our lifetimes. I mean, I hope that we do eventually, but I think that's more in the far future. So my lab has been focused on, could we search for life as we know it or as we don't know it in the solar system? And I think there's a lot of different criteria that we have to consider. One, for example, is what kind of life are we looking for? What assumptions are we making? Are we looking for life as we know it or life as we don't know it? If we're looking for life as we know it, then we have all the tools that have been developed on Earth for characterizing that life, such as DNA sequencing, for example. Maybe we can look for DNA on Mars. The challenge there is, unlike the Jurassic Park film, it's very likely that we can't get DNA that's tens of millions of years old, at least not on Earth. On Mars, where it's colder, we might be able to go further back in time, but even there are limitations due to space radiation and other degradation processes that might be at play. So probably no Jurassic Park, but we can look for life that's either alive or recently dead. Um, but that's also very specific. It's something that if life developed independently on Mars or on some other planet, Enceladus or Europa, perhaps in a subsurface ocean, then maybe it uses different building blocks. And so we have to start from what assumptions are we making about that life. And then there's a whole host of other considerations, like how abundant might that life be or have been? Would that target that biosignature that we are targeting, would that stick around until the present day? If life existed before, would it still stick around now? Would it be measurable, and at what abundance? And that gets us into all kinds of instrumentation questions. Is it sensitive enough? Is it specific enough to not have false positives, for example? So lots of different possibilities there.
Yeah, of course, and all the unknowns as well. Uh, just we're we only know us, so we never know what's out there.
At the same
Uh,
time, we look at meteorites and we find amino acids, we find nucleobases. And so it seems that the universe is a carbon chemistry factory. It's literally spitting out the building blocks of life in irradiated ices around stars and in other settings. And so uh, maybe life's totally different than us out there, but maybe some of the building blocks are a little bit similar because they're so common in the universe.
yeah, and um, so then I guess on the topic of the carbon chemistry factory, a question from another group mate, Vinny. He wanted to ask what your opinion is on life being based on different elements than carbon, like the silicon-based life.
Yeah, I think this is something, we've many of us have read science fiction stories about silicon-based life. And so that's a pretty common question. One of the things that's amazing about carbon is it's very versatile in forming bonds with other elements. And so that makes it very useful as a building block for life. Now, there are living organisms that are able to incorporate silicon into uh, into their bodies. There are plants that do this. There are diatoms. So there is a precedent for silicon in life as we know it. So maybe there certainly are biomolecules that involve silicon. That said, there my understanding is that there's a much restricted range of the potential bonds that can form. And so that means it's a less rich chemical space. So
Mhm.
if we were to postulate what that life might look like, I don't know. I'm not really. We might need to ask a chemist who's really good at silicon chemistry, um, but maybe it's robotic or something like that. Um, One interesting fact I learned recently: I have some colleagues who are studying chemistry in sulfuric acid, and I'm involved in. As a result of working with these colleagues, I'm involved in a mission to Venus in partnership with Rocket Lab. And we're working on a couple of future mission concepts as well. And one of the things that they have learned is it seems that there is a wider range of silicon bonds that might be stable in sulfuric acid relative to carbon. So, we are only scratching the surface with understanding chemistry in different solvents. So water is a polar solvent. It has a net dipole where the way charge is distributed in the molecule. And sulfuric acid is also polar, but it's a very different solvent than water. We clearly would think of it as an extreme environment. Venus is incredibly dry, way drier than any of the limits of life as we know it. It's incredibly, it doesn't even make sense to talk about pH. It would be off scale low, extremely acidic. And yet we think there's some interesting chemistry that can happen in that environment. And I have no idea if you would call it life. I don't think there's life on Venus, but if there is, that would be pretty amazing. What I do think might exist is some carbon chemistry, and maybe silicon, I don't know, in the clouds of Venus, and that would be really cool to understand better what is the limit of chemical complexity in sulfuric acid. And then there's missions to go to Titan, like Dragonfly, and study what is the nature of organics in a methane driven cycle. So I think there's a lot of things we don't know. And by going to these places and also by doing lab experiments, we're going to better understand the range of chemistry and possible biology that could exist in the universe.
Yeah, yeah, exciting stuff. And with chemistry, I'm not too familiar or knowledgeable on chemistry, but I know that tiny things make big differences sometimes. So uh, hopefully they're able to come up with some good stuff. Uh, so uh, I guess are there any current space instruments that or space instrument developments that you're willing and able to share?
Yeah, sure. We're working on a few different technologies. One is we've been working for a while on looking for life as we know it by targeting nucleic acids. And so the technologies to do that have changed dramatically over the last 15 years. So we started out with an approach to amplify DNA using PCR, targeting the most conserved gene regions in all known life to seek evidence of life that is ancestrally related to life on Earth, if that exists on Mars. And since then, we're now the current best technology for this is really single molecule nanopore sequencing, which uses biological nanopores embedded in a high resistance membrane and monitors the flow of ions through this pore as DNA is also being ratcheted through the pore by what's called a motor protein. And this is the kind of sequencing technology that was commercialized by Oxford Nanopore. It's really amazing. It's tiny. You can do it anywhere on the planet. You can do it in space. We've done it, we've done sequencing in the Arctic and in the Atacama Desert and in all kinds of places. And it's a phenomenal technology. It does have some complexity for use in space, which is preserving the biology, the reagents, the components that are required to make it work in space or to make it work anywhere, really. And also the preparation. If you get a sample and you want to extract the DNA, you have to extract the DNA using a variety of steps. And you also have to prepare the DNA, make a sequencing library. And so there's a lot of different automating that in a space instrument is really complex. And so that's the current challenge is automating that in a rigorous way to potentially look for evidence of nucleic acids in an unknown environment where you don't know exactly what the chemistry of the sediment or soil or regolith or whatever you're putting in. So that's one technology. Another is solid state nanopore. Same principle, measuring the flow of ions through a small pore, but now it's a synthetic pore. It's not a piece of biology. It's just a tiny hole. And the problem here is that the DNA can zip through so quickly that you have to measure very, very quickly. You have to have high bandwidth. And it means that with this technology, we can't really sequence DNA right now. That's kind of the holy grail. But what we can do is we can detect individual molecules. And a graduate student in my group, Jordan McCaig, recently published or has a preprint out on this, and it's in review right now. It's called Translation as a Biosignature. So we did a bunch of solid state nanopore measurements, and she's been able to show that by analyzing these, we can differentiate between DNA and RNA and protein fragments and ribosomes, which is the molecular machinery that translates RNA into protein and is common to all known life. So we think that might be another way to look for life in a little bit more agnostic way. We're not targeting nucleic acids per se, but we could look for long charged polymers at the single molecule level. And then a third technology is quantum electron tunneling devices. This is basically like a gold nanowire. We put it in bending. It's on a silicon chip. And the technology was developed by Masateru Taniguchi's lab at Osaka University. And when we bend and we break this gold nanowire, we now have a little drawbridge. And this drawbridge allows us to control the spacing between that gap. We can form an atomically sharp junction, like a gold atom and a gold atom, and we can control the spacing between those two atoms really precisely in the sub-nanometer regime. And single molecules can interact with that gap. We apply a voltage, there's electrons that jump across the gap. Now, normally there aren't very many electrons jumping, but when there's a molecule in between, that can change the conductance of that gap. Suddenly, there's a way for electrons to hop across it much more readily. And so we can read out the electronic structure of that molecule. And that allows us to do things like detect a single amino acid or detect the bases of DNA. And so in the future, this might provide a way to do that, not just for DNA or RNA, but for maybe a non-Earth-centric polymer, something that's a little bit different from life as we know it. So in general, we're trying to move from an approach that's specific to life as we know it to more agnostic and general to detect life as we don't know it.
Yeah, for sure. Yeah, it's yes, long chain polymers. And then I guess on the related to that, Isaac was here. Another in the group was asking or wanting to ask what possible what are some possible ways that a genome of extraterrestrial life could be different from ours if you've studied that.
Well, we don't know about any extraterrestrial genomes yet, but the project I mentioned that is we're trying to look for nucleic acids beyond Earth. We called it SETG, Search for Extraterrestrial Genomes. That was a project that got started. It was initiated by Gary Ruvkin at Massachusetts General Hospital, a geneticist, and he brought in an absolutely fantastic planetary scientist, Maria Zuber. And so both of them were were my mentors for many, many years when I was working on that project, when I was at MIT. And so I hope we get to fly that one day. That'd be really exciting. So if we get to sequence an extraterrestrial genome, what would it look like? Well, we know that it's probably going to be a microbe based on energy considerations and other things. It's likely to be a microbe. A microbe typically has a genome ranging from a few 100,000 bases to maybe 10 million bases. It typically has thousands of genes that encode proteins and also RNAs. And so we could look for those. We could look for those and we could compare them and we can see, is there evidence of conserved proteins? Is there evidence for conserved nucleic acid sequence? And so if we found that, then we could place those microbes on a tree of life. Now, this doesn't make sense to do this for, all possible destinations, but for Mars, we know that Mars and Earth had exchanged a lot of rocks due to large meteorite impacts that could knock material off of one planet and have it end up on another. And in this way, probably 1 billion tons of rock was transferred between the planets, mostly from Mars to Earth. And that's just the viable transfers. That's just the rocks that didn't get heated above 100 degrees C based on statistical approaches. And if you further restrict this to rocks that might have been transferred relatively quickly, those would be the ones that didn't spend a long time in space getting exposed to space radiation. You're going to get lower numbers, but still the chances are pretty significant that if there was life on one or the other planet, it could have been transferred to another world. And it seems to me that the most likely scenario, if this happened, was that life could have originated on Mars and been transferred to Earth. So the hypothesis we would like to test by sequencing a Martian genome, if we are so lucky one day, would be, are we Martian? And learning more about what that Martian life might look like today, and its similarities and differences from Earth life based on its genome. And of course, we want to study lots of other things if we find it, but I think a genome targeting extant life, maybe deep in the Martian subsurface, or maybe closer to the surface if we find just the right location, could be a great way to try to do that.
Yeah, hopefully we can continue research on Mars. I from what I understand, it's slowed down a little bit with some of the, is it Curiosity? Uh,
I mean, from my perspective, there's a lot going on Mars right now. We've got Curiosity, like you said, and we've got Perseverance, and there's Mars sample return coming up. We're gonna learn a lot from that. It's a very complicated mission, but hopefully we'll have some pristine Mars samples to look at. So,
yeah, that'll
um,
be. Um, yeah, it's super excited to see what comes from Mars. And
sounds good.
then
Sign me up.
yeah. All right. Well, I I appreciate your time. And I think that's all that we had worried about and kind of
Okay,
our
sounds great.
timeframe.
Corey, thank you so much. I appreciate your time too, and I hope everything goes well with the class. And good luck.
Yeah, thank you so much. Have a good day.